Okay, so this week 13, we are going to discuss networks and modules. We will start with some definitions of centralities, network centrality. That's for the statistics to define the importance of nodes in the network. Then we will look at a method called WGCNA. It's a very popular method before the single cell error, and people use that for co-expression analysis quite a lot. If you look at the citation for this method, maybe 1,000, I don't know how many, but it's very, very popular. So it's better to know the principle of this and this R package. So you can apply this to single cell or non-single cell expression data in order to compute the co-expression network. It's very, very useful. And then we will look at the community or modularity detection algorithm for network. Then we will move to single cell gene regulatory networks. And we will go through several methods such as Genie3. People use this to create single cell gene regulatory networks. And then the related classification with the tree or regression tree. And then at last, we will mention this multi-layer networks, especially when you have pseudo time for your cell, and at a different time point, you can have different networks. In that way, you create a multi-layer networks, and those networks will arranged according to the pseudo time and a different population of cells. If you have such a data structure, what you can do with this, okay? So very general framework for the functional network analysis. I just use very general examples. This is from a functional MRI data theory signals. So you collect the data from different points of the brain. Then this different location, physical location on your brain have given you a different signal across time. And those signals are correlated. If two of the signals are highly correlated, you can create a correlation between them and say, okay, this point and that point of the brain is highly connected because the signal is highly correlated. Another way, for multiple points in your brain, you collect data, and those data you can create between data points correlation, and if it's strong, then you put a high correlation, and it's weak, you put a low correlation coefficient. So it depends on this value, you create this correlation matrix, and you can use this as your foundation for produce a network between all the points in your sample location. You set up a cutoff, say, okay, if this coefficient have value greater than certain cutoff and is above the threshold, then you say, okay, these are connected significantly. Then you binarize your correlation matrix into a 0-1 matrix. 1 means the 2 points are connected. So in that case, this has become the adjacency matrix. It's a presentation of network. So with this adjacency matrix, you can see, okay, the points are linked, and some points, some nodes have multiple linkers and link with many different areas, other nodes, and this node may be the center and more important, the functionally. So that's the way, in general, people just collect the information from some kind of series data, and in this case, a time series. But in your experiment, it could be different samples, but you have to have some change, perturbation to the system, and look at the gene or other signal change, whether correlated together or not. In that way, you can create the correlation matrix and you build the networks. So that's the framework, okay? So we are going to use this framework to regular non-single cell gene expression matrix. In that case, you have multiple samples. Or later, we can use this to single cell data because you can create correlations between genes by looking at their expression across cell populations, right? Is that clear? So some terminologies. We call sometimes nodes and links in network science and in mathematical graph theory. It's called vertex and edges, but it's interchangeable. So if we have a network like this, 1, 2, 3, 4, 5 nodes, and then we can represent the network using adjacency matrix. In this case, this is a unweighted. So you just put any link have a weight as 1. And also this is unidirectional. So if you have a link between node 1 and 2, so you have 1 here, so that's a link. And also other way around, 2 and 1 are also linked. So this adjacency matrix is symmetric. In that case, you deal with unidirectional network. So we don't care is 1 regulated 2 or 2 regulated 1. It doesn't matter as long as they are linked. Of course, you can set up this as a directional if you think, okay, 2 give control and over 1, and then you can say, okay, this I keep as 1, and the other one around have 0. So you make this symmetric. So you can use this to represent undirected network. Also, you can put a weight between all this. So here, instead of 1 and zeros, and 1 becomes some values, real values, W between I and J linkers. Okay. I'm pretty sure everybody knows this. Okay, the first question we normally will have is to, once you're given a network, you want to look at the network and figure out which node is the most important one. Okay, important one is the subjective definition. What is importance? What is of importance? So we do need some measurement, and the group of measurement called centrality. The simplest centrality is degree. It's how many nodes, how many linkers around this node. Okay, normally we think it's a highly weighted. Okay, high degree node is more important because they have more partners, many links around it. So if you knock this gene out, it's more likely this region of networking will collapse because there's no central gene and make link with other products of the genes maybe. So that's the most naive way, and this called the degree centrality. In this graph, you see the purple nodes have a degree number 4, because 1, 2, 3, 4 have 4 different nodes linked with this. Well, this is a very naive definition of importance. So nothing wrong with it, because a lot of other centrality are correlated with the degree. When you have a higher degree, normally you have many other centrality measurements also high, but not always true. And sometimes you have 2 nodes have same degree, but because of topology position of one of them, you want to have a different ways to measure the importance, whether these 2 nodes has equal degrees, whether one of them is more important than another. So that's why people invented a lot of different ways to do measurements of centrality. And for instance, this blue one. Okay, blue one only have 2 links. Looks not very important, but however, functionally may be very important because they are in the bridging point of 2 large modules. You have one module here and another densely connected, but this is a bridge. If you break that down, you have a global dissociation between modules, and maybe not important for the within module functionality, but you broke the bridge. So you want to define something and to say, okay, this particular one, although it's not highly connected with any other nodes, degree is low, but it's topologically important. So we want to have some this kind of different ways to compute centrality. And for me, so the network science is very intuitively sound, and oftentimes you can invent your own centrality definition. If you know the problem, you want to capture some of the signature of the nodes in the network, you can define your own centrality very easily. I'll just show you several examples and to illustrate the point. One of this is called closeness centrality. Okay, what the definition of this? So closeness centrality is the degree of all other nodes connected with it, and you sum them up, and then 1 over this sum value. For instance, you want to compute the centrality of A. Well, you will look at the distance. Okay, so this is the distance, not a degree. You look at the distance from all other nodes linked to A. So for B, you have 1 step. If you from C to A, you have 2 steps because every step here is 1. In that case, you have a distance from A to B is 1. From A to C is 2. From D is 3. To 4 is E. So you sum them up and then 1 over it. So how can you make closeness centrality large? You see, C has the largest centrality closeness because it's in the center of this so-called network. But here is not a network, it's a straight line linked with all the nodes. But the same idea, if you are in the center of your network, you are mostly likely to have a high closeness centrality. Okay, your degree don't have to be higher because you see C and BD have same degree. You all have 2 partners around it, but the C, because it's the middle of the network, measured by its distance to all other networks, to other nodes. So that's a very simple way to compute this, right? So you need to know how to compute the shortest path from one node to another. Just look at the focal node to all other nodes in distance. Another way is to call the harmonic centrality, and it's very similar to the closeness centrality, but the equation is slightly different. You 1 over the distance first, then sum them. So this is called harmonic sum, harmonic mean, basically. Okay, so just slightly change of your equation. And now the harmonic centrality for this node here is the distance to all other nodes. So you have a distance to this node is 1, to this 2 nodes are 2, this node 3 steps, this 4. So you 1 over each of the distance, then sum them, and then you have 2.5. Okay, so these two are called disconnected, so we treat them differently. Here, all the centrality is computed by the so-called connected components. We only consider those connected components. Here you have another separated component. Questions? Degree? Oh, okay. Okay, so here is a funny part of this. First, we will look at what is the power of adjacency matrix. Okay, in this graph, we have 6 nodes. Then its adjacency matrix, here use M as a notation, and M is 6 by 6 matrix. Is this a directional or undirectional? It's a directional. The reason is not symmetric. So you see, okay, here we have from 1 to 2. So this is node 1, this is node 2, 3. And we have 1 here that indicate you can go from 2 to 1 this direction, but you don't have a link from 2, no link. Okay, so this 0. But this can be generalized. The same principle apply to both directional and directional. So here we are going to do a power. What that means is you will multiply M with M. Okay, so that's a squared of matrix. Or if you have a power to 3, then you have M times M times M, or power N, and then you have M power N times. So I want to show you what that means and what the result will be if you do this. So first we are going to use squared. Okay, so that will be a matrix of this 6 by 6, M times matrix 6 by 6. So the output of this will still be a 6 by 6 matrix. Okay, so you know from day 1 you do the computation of a matrix multiplication. You know, multiplicate one matrix with another, the dimension, and then you can compute that. The way I showed you is to put, remember, I show you put the first matrix here. Okay, so that's your 6 by 6. You put another matrix here, 6 by 6. In their dimension works, so again you have this. That's 6 by 6 matrix. Okay, so that's matrix multiplication. And how can you compute the values in there? And you get one row out and one column out. And then to do this 1 by 1 and the corresponding values, multiplication and sum. So this position will be your I and J position in the product of your matrix. So you can do this. And in this case, if you have power 2, you get that's the value of your multiplication. And we reset this diagonal position to 0 because we don't want to have the link from the node itself, self loop link. but you can keep them there. Okay, let's see what that means here. And now this value, remember it was 1, and that value was 0 before multiplication of m itself, but now become 2. So that's the meaning of this is when you have 2 here, the meaning of this value 2 here is how many ways you can go from a node 1 to 2 by 2 steps. Okay, so you see from 1 to 2, we don't have origin, it's not linked. But now when you take a power to the 2 for the adjacent matrix, now it's become linked. And this link tells you from node 1 to node 2, how many ways, how many different distinct paths you can go from 1 to 2 by 2 steps. Okay, so you see, we have one way to go from 1 to 3 first, then go to 2. So that's a 2-step link. So that's one way. Another way is so you cannot go from 1 to 2 directly, but you can go 1 to 4 first, and then go to 2. So that's a 2-way I can identify it from this directed matrix, directed graph. So this 2 means that. Let's see, we have a large number 3. So what that mean? This means if you have 3 different ways using 2 steps and go from 4, I think it's from 1 to 4. Let's see if this is correct. Okay, so this number indicate I have I have 3 different ways from node 4 to 1 by 2 steps. Okay, the first way is right. So that's my first way. Another way is the third way. Okay, so you can see, okay, this power of adjacent matrix indicate an indirect connection between 2 nodes by number of the power, which is how many steps you will use. If it's 1, that is adjacent matrix itself, with how many ways you can go from one node to another by 1 step. That's a direct link. And this 2 power 2 gave you the 2 steps link. How about power 3? So you can do the 3 that indicate indirect link from one node to another by 3 steps. 4 by 4 steps. So you can generalize this concept and compute an indirect link between 2 nodes by n numbers of steps. Okay. Now what we are going to do is to do the overall connectivity between 2 nodes, any given nodes from I to J. Consider all these possible links by direct link plus 2-step links, by 3-step links, by n-step links, infinity numbers. So if I sum them together, I get a global connectivity between 2 nodes, not only by direct link, but also by all other possible steps links. So that will be M, which is 1 plus M2 plus M3 and then a certain number we stopped because it will converge. And so this gave you a global, I think we should have a good way to call this is overall connectivity between any two nodes. Okay. And also this concept is very important. You can think any observation you build your network from your sample. So this network could be your observation. That's your observed. Your observation contains direct link between two components and also many other indirect link. Okay, what you see in your sample, maybe not adjacent matrix. So you observe a combination of all the possibles. So if you have a way to do the decomposition, if you do the reverse of this, instead of take power, take exponential of matrix, probably you can decompose and reconstruct real adjacency matrix between nodes from observation. Okay, so you have to think about it the other way around. So that's computed directly linked adjacent matrix, maybe a true connections between genes. Okay, I call it intrinsic link between genes, right? What you observed is not, it's combinations of all these, all possible links through other ways, through other genes, third-party genes. But if you have observed matrix, which is sum of this, and reconstruct M, you probably can get the intrinsic link between genes. But that's out of topic of here, and there are a lot of numeric computational problems to do this because you content noise, maybe not converged. It's difficult to do the reverse. But here we consider forward. Okay, so if we have an adjacent matrix, we know the powers of this means that, and in this index called the cost index, use this concept to look at the importance of the node. Okay, so what is this? It first compute all this power matrix, take powers, and from 1, 2, 3 to the infinity, and then sum them up and look at this summed. So you have adjacency matrix, and then you construct this new matrix by taking them together, and then look at each node distance, okay, to all the other nodes. Yeah, basically, so you look at its link with other nodes. So then you compute this so-called causal index for each of the node in the network. It not only consider direct link, but also consider the possible other links between nodes and give the weight for each of these nodes at the end. Okay, here is real network examples to show values of different centrality computed for all the nodes in this network, and their relative value was indicated by the color. And the topology and number of genes are the same here. Well, if you look at the degree and you found those genes have a higher degree here, but if you compute closeness centrality, will this part of the nodes have a highest value. And if you look at harmonic centrality, will this group have a highest value, and the cos centrality indicate this part of the gene have the highest value. So you see, they are not unrelated. There's some kind of correlation. This is part of the highest degree. You have a highest closeness centrality, and also here. But it depends on your algorithm. Your centrality may indicate some part of this network. Some group of nodes are more important than others. Okay, so you can combine all different measurements for any of the nodes. So in this case, every node has 4 values because we compute degree centrality, closeness centrality, harmonic centrality, and the cos index for them. And if you look at this gene, it's always lower value, right? Because it's in the edge of the network. Maybe it's not important, or at least by our indication of those parameters, this gene is not important. But some genes may be important in this way, or more important in that measurement, but not in that. So give you a multi-factorial view of the importance of topology position of those nodes. Okay, so in real application, if you can apply this to some of the established network, you may have a better view of the importance of nodes in the network. As I said, depends on your application. You may want to invent or create your own centrality index. And this is a relatively new paper, and people just pop up a new index. This is called leverage centrality. They use, okay, so here, N, what's Ni? So it's average over all neighbors. So you look at one of node. Okay, so for example, you are working on this node I, and then you look at its neighbors, all as neighbors, direct linked neighbors, and you look at its neighbors degree. You use node I's degree minus all your connected partners degree, and somehow normalizes this. So if this node have many partners, but your partners have very few other partners, then the difference is large. Then this index leverage centrality will be large for node I. Otherwise, if you're in the middle of this, if you I here, then your partners also have many connectors. So you are in the middle of a lot of popular nodes. So your leverage centrality will not so high. So this one will certainly give high value for those kind of nodes, because they have a lot of partners, but their partners isolated. They don't have any partners. So you see this value. So you can use this to discover some structure like this. So it's called dissociative network. If you have a lot of nodes with a high value of this, so your network will probably have this kind of structure compared to this more homogenized topology. Okay, we are done with this centrality, different centralities computation part. Any questions? Online students? I just assume you are all good. Okay, we move to WGCNA. So the full name is called Weighted Gene Co-Expression Network Analysis. Its most popular one is R package for co-expression network. You see this all the time. It doesn't have to be the best one, but one of the most popular, and there are some features make this very stable, and people want to use it to find co-express modules. See, I want to look at whether these 20 genes in the same cluster, same module, they always upregulated, downregulated at the same time. And then another group gene also do this. So it's very useful for those downstream analysis. Once you have those modules, these 50 genes always regulate together in a cell, in this sample. Another co-express. Then once you have gene set, you can do a lot. You can do annotation. You can see, okay, whether mutation SNPs are located more abundantly in those group of genes and associated with those disease. And you can look at whether those genes share the same biomarkers or regulate under the same regulatory factors. Okay, but those are the downstream, but we are mathematically or methodologically, we focus on how are you going to construct this gene co-expression network to support, to identify those modules. Those modules are the combinations, numbers of genes, different genes. And then you can look at pathway enrichment. Okay, so we will focus on these methods. Developed by Steve Horowitz, and his UCLA professor, and they published early and occupied market basically. And himself is working on the molecular clock for aging using methylation patterns. Okay, so WGCNA is a product of pre-single cell error, and at that time, people just collect samples from one condition and then look at another condition. So mostly you have two levels. Okay, in that case, you can see, okay, a group of gene upregulated in treatment group compared to untreated or control group. So you always have some kind of this. Then you look at control group with his group and look at some genes. So those red genes are co-upregulated. And there's some degree, then you can see, okay, they have correlation coefficient across maybe samples. And then those blue ones, although correlated but not changed, so another group. And another, so called downregulated. So you can establish this correlation between genes, but within the boundary of this so-called clusters, then the correlation is weak. So you have this correlation-based matrix. So that's our starting point. And I don't trust those because normally we don't have enough samples. Okay, you have two groups, and each group you may have replicates some example biological replicates, and then you have some kind of variation. But you see this correlation, a lot of correlation will be false positive, I believe. And very noise, and the correlation itself is not a good measurement to do this. And also because you have numbers of the sample conditions, you cannot have a lot unless you have many different time points. For example, you have a gradient-based correlation, regression-based analysis. You can do this, but in general, I don't trust those early stage of those co-expression analysis, because you generate a lot of pseudo or false positive signals. Well, at that time, that's what we can do. You have those gene by gene correlation matrix. That's the first step. Then the framework will help you to construct a network by several steps, filtering and enhance the signal and try to get rid of those noise. And then you have module detection. This is using clustering, tree-based clustering algorithm. Then the next step is to use this gene sets in the modules to do downstream enrichment analysis. So now we are going to look at step one, how this network is constructed and filtered in order to smooth the signal and try very hard to get rid of this potential false positive signals, noise, basically. Okay, first, you can take. Okay, so this method is not very good to deal with negative correlation. And we have trouble to understand negative correlation, actually. So if you have a gene and say negatively correlated with another gene, and this gene will also positively regulated with a lot of other genes normally, and this gene positively regulated with other groups. So you have these two big clusters of genes. Within the cluster, they all positively regulated to each other, correlated. How can you establish a negative correlation between two members of this? A negative correlation. Well, the other genes still have some weak positive correlation. Somehow it's difficult to, I have trouble to understand negative correlation between co-expression gene network. In this case, they deal with this situation. Once you have some negative values, you can either take an absolute value, just ignore it's not there, and make an unsigned correlation coefficient. So here S is called similarity measurement. There are correlation coefficient, whatever the way you get it. Another way is to do this transformation that will transform your if the correlation is -1, and then this becomes 0, and if it's 1, become 2. So it's just make your similarity measurements no negative values. Okay, after this transformation, your values will be between 0 and 2. Basically, you move everything up by one step. Okay, so WGCNA just uses different ways to try to surpass this, not deal with negative value at all. So your similarity matrix input will be converted into non-negative matrix. Then they set up a cutoff. See, I want to have a cutoff hard threshold. Let's see. You have to have similarity value greater than something, and then these two genes are linked. You can select this. Or you can do a transformation. So I take power of beta. Basically, I transform my values from 0 to 2 values from here using this power. What that means is if you have a value, say 0.9, and then you take beta equals 2, and basically you square it, it becomes 0.81. If you have a weak connection, 0.2, for example, you square it, 0.04. Okay. So you make this weak link become much weaker. Slightly weak becomes slightly weak. And if you have 1, then unchanged. If you have 1.2, you make it slightly larger. If you have 1.9 squared, it become larger. So you just spend, you just push those weak link close to 0. And any strong link becomes stronger. So this just makes signal stronger and make weak signal weaker. Okay, nothing special. Well, you spread them out, the values, by taking this power and then reset a cutoff, and then somehow you have an option to adjust the beta values. Although you still use some kind of cutoff. Anyway, so we start by make the matrix become non-negative, and then set a cutoff, make it become sparse, and become a network. Because after you treat this using cutoff, it becomes a 1, 0. Okay. Either genes are linked or not, then you have something like this. Then the next step is the key, and you do a so-called, you transform your matrix, similarity matrix. What was the original was correlation matrix between genes, and replace it with a so-called topological overlap matrix. It's HOM. So you start from your similarity matrix after some cutoff, and then build the network. And you don't use this as an adjacency matrix directly, but you are going to look at the links between all the genes and use to replace this network using this topological overlap matrix. Okay. So now we need to compute, okay, what is topological overlap matrix? If you consider 0, that's it's a parameter called so this is called the power rank 1 topology logical overlap matrix. If that's 0, that's indicate there's nothing changed. You just use the adjacency matrix you obtained from previous step. But if you have 1 here, indicated we will consider each node one-step network neighbors to construct this topological overlap matrix. Okay, so here, WGCNA use topological overlap measure as a proximity. And gave you a weighted network, okay? So this this matrix or this measure look at adjacence of 2 genes. So you compute the link between 2 genes and use these measurements to indicate whether they are strongly linked or not. You look at the neighbor 2 genes, they are linked, but also consider the connect strengths of these 2 genes shared with all other genes. Okay, if these 2 links shared with a lot of other genes, but this so-called neighbor other genes overlap a lot, then their link is strong. The idea is like this, okay? if you have two genes, original link may be strongly correlated, but their neighbors, their neighborhood, not overlap. They're far, maybe only overlap a little. Then this topology measurement will recompute and give you weaker link between these two genes. Okay, so basically it will merge those genes shared a lot of neighbors. So how this will be done? Okay. Now we have a definition, the adjacency matrix we get from the previous step, and Aij indicates the link between gene I to J. A can be weighted or unweighted, it will be 0 or 1, okay? Then if you consider two nodes, see, gene I and J, if they connect to the same group of other nodes, then they have a high topology overlap. So you need to look at those two nodes and look at their neighbors. Okay, when we consider Tom1 indicate we only look at one step neighbors. If two, then we look at two-step neighbors, okay, to expand this neighborhood for each of these genes, I and J. So Steve Horvath, the professor, borrowed this idea from a previous publication published in Science in 2002 from a Bassi group. So at that time, they proposed this idea called let's see what's the name of this, topology overlap. So this concept was not new. The idea of this topological overlap is for any two given nodes, I and J, its topological overlap is the shared neighbors of I and J. So Jn denotes the number of nodes which shared between I and J as a neighbor. Okay, they are linked and then normalized by this degree of minimum degree of I and J, either of them. Okay, somehow normalized. So if you have two nodes, they have a lot of shared members, then you have high topology overlap. That's a definition. And this rule applies to weighted network as well. Then in this paper, they define this. If it's weighted, when you look at the two nodes, these two nodes are the different nodes. If they're the same, then the topology overlap is 1. This I and J are different nodes, and then L is the sum of all their neighbors. Shared neighbor, I believe. Okay, the weight between their neighbors. Okay. Okay, so this U is the third party of the nodes, which is not I and J itself, it's all other nodes. Their weighted link between them and sum them is not only count how many nodes are shared, but also count how strongly linked with those neighbors. And then you have this, their direct link between them, and then minimize this degree. So there's a definition like this. Okay, so if you have unweighted, network, so then you don't use the degree, the sum of degree. You use the number of shared neighbors. So here is I and J and their one-step neighbors, and how many of them are shared. So that's the number of shared neighbors minus plus whether they're linked. And then you have minimum number of shared neighbors plus 1 minus. So that's a definition of for the unweighted network, the topological overlap measurement. Okay, let's take a break and then we come back to see 1 or 2 examples of this, how this is computed, for example. In the case D, the topology overlap measurements between 1 and 2 nodes is this. Okay. 5 minutes break. Let's come back from 6:11. Okay, I'm going to use this example to show you the computation of topological overlap measurement. So you have node 1 here, node 2 here. They are not directly linked. So A between 1 and 2 is 0. Okay. They are not linked, but they are undirectly linked with this shared, this shared, something like this example. So now we are considering the topological overlap between node 1 and 2. You have to do this for all different nodes pairwise to recompute the adjacency matrix. Here, you certainly have adjacency matrix, right? Okay. Let's see. So you have adjacency matrix for this network. So you want to recompute, so all the values here is I12, value Bij in there, and you want to transform this into topological topology measurement. So for each of pairs of this Aij, we want to transform into Mij. And for example, here I'm considering node 1 and 2, but you have to do all other nodes. Well, the link between these two, 1 and 2, is 0. No link between them. However, I want to compute this value between M1 and 2. So this M1 and 2, by definition, is our the shared number of shared nodes. Or how many nodes they are shared? So we have 1, 2, 3, 4 in here. So this is first. This is 1. This 1. This 1. This 1. So you have a 4 number of shared nodes plus Aij plus definition. Here is degree for 1, degree for 1, degree for 2, and the smallest minimum plus 1 minus Aij. So number of shared is 4, and their degree is 5 each. Okay, A degree have 5, so this is 5. And 5 minimum. Plus 1. Yeah, J is 0. So in this case, our value is 4 over 6. Is that right? So that's a degree of, so one degree has 1, 2, 3, 4, 5. Two have 1, 2, 3, 4, 5. They are
Okay,
symmetric, so
okay.
they are same. So you just found the smallest.
Okay, so it's not just the shared ones, it's all of the connections.
This is their partners, and these are shared ones.
The top is the shared.
Or
and
shared.
the
Yeah,
dominant.
four shared. Okay, so you compute this and replace your original adjacent matrix with this field values. Okay, you can also consider not only one-step neighbors, but also two-step neighbors or three-step neighbors. And here illustrate the adjacent matrix, which generalize Tom 0. This is adjacent matrix, original matrix looks like this. Okay. Looks like this. Then if you transform the matrix to topology overlap matrix, then by consider just one-step neighbors, then you somehow smooth out the signals. If you say I take 2 steps of the neighbors and you give 1 step more smoothing of your signal. And step 3. Okay. So this Tom matrix compared to your original adjacent matrix is more smooth, and you get rid of a lot of potential noise because you not look at whether directly linked or not, how strong they are. You also look at their relative neighborhoods. So topological overlap to help you to get rid of noise and to allow you to cover those potential links and give you the large chunk of those blocks. This will be used in the next step to do this tree-based topology module detection. Look at their similarity, rearrange all these nodes, and then by the similarity, they put it together, and they're so you create those blocks. These blocks then report as the gene sets. Now here to you, adjacent matrix give you a lot of those these significant blocks information will you transform into Tom matrix, improve the situation. And then if you use this more generalized, because you have you consider multi steps of the neighborhood, and you get much clear separation of this gene modules. Okay, so this is the original, just one-step neighbors. But you consider two-step neighbors, you give much better separable module or group of genes and much clear signals here. Okay, so the downstream analysis, once you have this matrix, the downstream analysis will be straightforward because people have this method to from a distance matrix. Of course, this distance matrix is the topological similarity matrix, just one over it, one minus it. You have this distance matrix, then plug in all this, will establish tree-based hierarchical clustering algorithms. You can get this modules. Okay, that's what the WGCN will report. It will report this tree structure, and also at a different cutoff, you have this merged matrix gene sets. Each color represents one cluster, and within this cluster, maybe 100 genes, 200 genes, 20 genes, depends on the size of those clusters you get. And then if you have some other measurement for, in this case, for example, the clinical trials, you have different other measurements for your sample. You can link with those parameters from external information. See what genes in which cluster is mostly positively correlated with these features, other variables which are not, to give you this downstream analysis. Okay, but those are not so special, and it's just very general correlation. Okay, here we have this definition of those hub nodes, and for example, like this one is in the middle of different modules. So here is the module. Module means you have a highly connected subnetwork, and within the network, those nodes are densely connected. Well, between different nodes, you can have intermodal hubs. And you can also identify the intramodal. So the hubs is within those modules. To identify this community or modules, one of the most popular algorithms is sort of called Lovin, and this is to compute an index called modularity index Q, and look at adjacent matrix and the so some kind of within the module, and with you have to consider the membership, see I and J as an indicator. Okay. Yeah, the overall idea is you have to consider whether these two. Okay, so you per set, so you assign all the nodes into modules. So you have indicators. Node 1 is module 1. Node 2 is module 3. Node 3 is module 1. So you have an indicator of whether which community each of nodes belongs to. Then you start to compute within module distance, or some kind of measurements between nodes distance, and within and between modules. So you want to change those indicators so you can reach some optimization level, which within the module distance is small, then between module distance is large overall. So that's basically the idea. The optimization procedure is relatively expensive computationally. So once you see this, it will take some time to compute, because you have to assign this membership for each node and to guarantee they're within the module distance between nodes is small, and then once you cross the module, then the distance is large. So there different algorithms were developed in order to do this. And in this case, it's comparison between co-expression gene regulatory modules. You look at the different conditions, say whether those co-expression networks can have a different those network modules assignment. Okay. Now we are going to move to single cell related gene regulatory networks. And as we know, we now have a single cell expression matrix. Now we have information for many cells. Okay, so this information per se already increased the number of samples to the n. But remember, before, even you have a multiplication, the replication of different conditions, you normally don't have over 10 samples for each condition. Nobody can do a lot of replicates. But in this case, we have a large number of n, okay, thousands of cells. If you think each cell is an independent trials, then in this case, you have n to the thousands of samples. So the correlation you will estimate across genes, between genes, will be more informative compared to only have 7 or 10 points. But here you have thousands of them to look at the correlation between genes. Of course, we still have the problem of the dropout. If you have a lot of zeros, okay, you have to deal with it. And hopefully your correlation measurement will not so sensitive to those. Okay, so now we are going to use single cell data to construct the regulatory network. Let's see what's the advantage of those. First, we can do this cell type specific, because you can group cells into population, and each population represents different cell types, and you build gene regulatory network for different cell types. But before, because of bulk samples, you have a mixture of signal from the different cell types, different tissues. You have a global or average correlation between genes. So for example, gene 5 and 4 are co-regulated, or one regulates another. But in this single cell world, you can see, okay, this only happened in this purple cell type. Okay, it wasn't in the orange, wasn't in the green population. So that's make all the network inference from single cell data become cell type specific. That's pretty important, right? And also, single cell data itself contains a variability. Well, in your traditional treatment, you have 2 conditions. You only have high, low, low, high. So that's only 2 conditions. It's not a continuous. Well, single cell data itself, because cell at different stage, even though you have a homogeneous population, cells always have some level of development. So you have this variability. So you can use this variability to look at the co-expression or co-regulation between genes to give you strong signals compared to those traditional population-based analysis with a few samples. And you can collect cells from multiple time points. You can build a pseudo time, and then you can look at this variability across those time points. Okay, so this review paper just show you some kind of options you can do. First, you can start from the samples and just treat as and build a network directly, right? Or you can separate them into clusters. Basically, this is cell type, and you build network for different type of cells. And you can look at the difference between them. Or you can arrange cells into trajectory, use pseudo time, and then you have a dynamic network to construct it along the time. Profile specific. I don't know what's this. Profile specific network. SYNIC. I don't remember what that exactly means. Profile specific. We will mention the SYNIC, which is one of the methods we use. To build a gene regulatory network, you can use different methods, including this review paper. So this one, for example, using information theory, and so this one is random forest. This is ODE, ordinary differential equations. So this require the time, okay, to arrange the cells based on the time. This one also, for instance, so there are different algorithms you can use to infer the dynamics of network. So again, that's a pseudo time related. You order your cells, and then to build maybe models and to see the change. Otherwise, if you don't have that information, you will lose this dynamic information. This part is more active for the blue cells. And this part is more active regarding to the red cells. If you don't have that information, you just build a global networks, and you don't have that dynamic parameters to show the change of that. Okay, some of these algorithms, and we will mention GENE3, and the SYNIC is based on the GENE3, and plug in some transcription factor database and target genes. SYNIC itself is not an algorithm, it's just a combinations of different components. And this is information-based, so-called neutral information and partial neutral information. This use time series data. This also use time series data. So this method has to be applied to very specific situation. You have a certain reliable time assignment for the cells. And because we have many different algorithms to for build the gene regulatory network. So there's a benchmarking algorithm to compare all this network inference algorithm and give a overall comparison between them using some kind of a precision measurement to see how good each algorithm is, and also they consider the running time. It's a pretty hard project because you have to consider all different algorithms, and some are R package, some Python, some are Julia. So you have to have a unified platform and using the same input data set, time them. Difficult to measure the accuracy of the output. What they consider all these available algorithms. So this is just part of the table, and then look at the different measurements and give the estimation of the quality of the based on those measurements. Yeah, 3 dots is either 30 minutes, and 1 black dot, by which only need 1 minute. Well, this is a systemic analysis, but the problem with this survey study is they use very small gene regulatory network as a ground truth. So for example, this is the largest data set they use. It's probably only contain less than 30 genes. Because some of these algorithms just cannot run efficiently enough for the large gene regulatory network. So they have to adapt this benchmarking study with a small network as a ground truth to test all the algorithms. So, my point is, in real applications, you don't have you don't have to take this one paper suggestions. Okay, this is the best algorithm I use. You have to be very adaptive. You don't know. You have to know the algorithm itself, see whether it will apply to your condition, and you need to know the complexity of the algorithm, see whether it will apply to thousands of genes network construction. So in that case, we have 6,000 genes in a single cell. Then you adjacent the matrix will be 6,000 by 6,000. Whether some of these algorithms will be able to finish in a reasonable time, we don't know. Okay, some of this will be very slow. Okay, so here is the summary of this study. I don't know exactly which one they provide to be a reasonable good. It's hard to tell, right, from this table. So these two are not too bad. Okay, gene 3 is not designed for single cell, but people found in the test one of the reasonable option if you can use it. It was designed for co-expression analysis for this gene regulatory network construction. So the paper was published in 2009, and it's one of the best performance in the test. It's called the DREAMM-4, DREAMM-5 test. Basically, they are the organization set up a ground truth network and gave the raw data to all the competitors and see which algorithm will recover the original network, and the best one will be the top performer, and gene 3 is the winner of this test. So the underlying algorithm of this is called the random forest. Okay, so here they first generated so-called learning samples of the input of the pairs. yeah, it looks like they use some kind of a subsampling strategy to do this tree, and at the end they combine those, assemble those information and give the rank of gene list. I'm not very familiar with the internal algorithm of this. So they call this learning samples, and to use part of this as input gene and some as output gene, and use input gene to predict each of the output genes expression, and see what's the weight for each of the input gene as a predictor to give the prediction of output gene. So you learn this structure and assign the weight for all the genes to this blue genes, okay, and make a link between them, and then they move to another until they loop through all the genes in the data set. So the underlying principle is based on the tree, and this tree can be used, the decision tree algorithm can be used to do classification and regression. So we all know this decision tree is a binary tree, depends on some of these yes or no answers, binary answers. And then you can use these parameters, like height, weight here, to classify your samples into subgroups. And if you have a training data, so you know the category of this, then you can use the training data to reset, to help you to adjust to each of this cutoff for this yes, no answer. And then at the end, you will try to partition your samples and put them into different categories that fit your original assignment of the cluster of the training data. Then you set up your binary tree. You can apply to the unknown data, the new data sets. And you can represent this tree at this higher dimensional space, and you use a separation of the first parameters to separate samples into this group, that group, and then another group you have another separator variable and separate it to different categories, and those values will be assigned and put into a different group, either categorical, or when you're in the regression case, you want to have a continuous values in there. But that's just representation of different input space, partition samples. But yeah, so internally you have this tree construction algorithm, then you have a different pruning algorithm, then you set up the stop criteria, and when I to stop the learning tree. Anyway, so you can also apply this to do the regression. Regression, you can consider you bin those categories into very small bin, and each have its continuous value. So there's no substantial difference between those, but you can just use this for the regression purpose. I believe Gene3 are using this as a regression method to regress all the other genes contribution to explain each gene test. So start to test gene 1 and look at gene from 2 to P, then you consider P minus 1 gene's contribution to explain the gene 1, then use the tree regression to predict the results. And then the algorithm moved to the next gene and use all other genes which not to the next gene and, as a predictor to predict the expression of next gene. So it loops through all the genes to create all these small trees. Then they combine the weight and then rank genes and to get the final network of the regulation. Well, as I said, SENIC itself is not a tree construction or network construction algorithm. It's just a borrow Gene3 as a first step to create co-expression or gene regulatory network first, and then they do search for the transcription factor and the target database. And if this correlation between transcription factor and its target is supported by the network constructed using Gene3, it will keep that relationship in the downstream analysis. Then they use some other algorithm to do this enrichment analysis of functions. But this paper has been widely cited. A lot of people use it, especially in the experimental validation. I believe because this database give you the support for relationship between transcription factors and their targets, and also this binding motifs. So give you strong support for the experimental results if you found some relationship using this method. So that's why it's highly cited. Another paper highly cited. So this is one of the example of SENIC. Apply this pipeline, identify those relationship, and then look at the motif binding. Okay. So this is another using partial information decomposition. They implement in Julia, and then they claim that if they are not using Julia, it may be slower in other languages. Anyway, so this is one of the methods to use the information theory to construct a gene regulatory network. It will consider not just directly positive, negative, monotonically regulated genes. If you have two genes, it's go up in some time point and go down at the same time. So this information-based measurement will still capture those relationship between two genes. Okay, so that's information theory, and you consider the different contributions of so it not only consider a pair of genes, also the other gene contributions. So that's why it's called partial information decomposition, to decompose those information measurements into different components. There are a very specific way to do this. Okay, I see this S chord basically it's need this pseudo time information to be in order to model the regulation process using differential equations. And scrap, so there's another package I try to infer the causal network from single cell data. And also it requires time points for the cells. And it learns both linear, nonlinear causality, recover causal interactives. Okay, so I at that time is still in the bioRxiv manuscript, but I don't know what's the development of the recent of this citation situation of this paper. Well, okay, at last we are going to move to have a quick go over to this multi-layer network method. So here we will consider whether their modularity change. Modularity is a community, okay, we detect from a network. And in this case, we will consider whether those membership will change along the time. For example, in this case, in the early stage, in this black and the blue community are two separate communities, but at a later stage, they become one merged community. So people are thinking about whether we have any method we can robustly identify those dynamic change of community structure in network. And look at the paper published in this journal. It's not a biology journal at all. So it's definitely a problem out of the biology field. Many network study field people are interested in this kind of problems, but certainly we can borrow whether the algorithm was designed to do social network modularity detection or not, or other methods. We certainly can borrow them from the other domain of knowledge applied back to this multi-layer network method to biology. Well, we don't have a good application yet. I don't see any kind of this application. But I believe there's something in there. You see, when you have the multi-layer and community quality measurement, this equation change. You remember that it was like this, and then you have another parameters adding because of the multi-layer problem. So what we want to detect? We want to detect whether there's a network switching or rewiring events happened. For example, this one was part of this and become the blue modules. So whether we have those, any kind of algorithm can detect this, it will be really good. And we can build this network right now using different population of the cells in different time points. Okay, right. Again, I don't think this paper would develop this method would apply to single cell data, but certainly we can borrow this. If you Google or search time varying graph, you will have a large number of literatures and network science, mathematics, topology, and people study this. And again, they want to look at this, the change of the interaction between genes, modularity form or across different time points. So there certainly have those algorithms we can borrow, but we don't have a good applications in single cell data yet. But last, this one is using non-negative matrix factorization, but consider multi-layer networks and try to identify those networks which modules which consistent cross time points, or and also which nodes, which gene in those part of the network. So this is one of the algorithms that slightly towards the way I just talked about, using multi-layer networks to look at gene regulatory module, either conserved cross time points or changed. Yeah, but so far I don't see many applications of this yet. We will see. But anyway, those are the most advanced algorithms that people are still considering. I believe it's still developing. Yeah, that's all we want to share. And of course, beyond those topics, we also mentioned about spatial transcriptome recently developed. What else? Spatial. Yes. We talk about pseudotime. We talked spatial. We talked about the ATAC-seq and multi-module. Yeah. So basically, I covered. I tried to cover all the topics we have already. We don't have a spatial transcriptome lecture yet. But if we have one more week, I should have that as well. Okay, that's all. And please let me know if you have any questions and comments. And next week we will have your presentation, and I will also give you some time to give me the feedback using the website for this course. So any place I should improve, or for the beneficial of the future students. The direction. And also, as I said, we will have a summer course on the quantum computing. So that's just one credit course. You will sit here and listen to why the quantum computing will be important, what your contribution could be. Okay. All right. Thank you. And see you on Thursday.